All right, good afternoon. Welcome back to the IPPS. Our next session will be on developments in gene banks, very important topic. So I just wanted to take a moment to remind everybody of what is being shown on the screen, that there is a raffle that's taking place. If you want to win a multi-spec and Cali-spec from PhotoSync, make sure to sign up for the raffle. And we will be giving that away this evening at the banquet. So if you're walking around, you might see a little piece of paper like this. Anytime you can scan this and sign up to win. Okay, I'm going to pass the microphone on to Peter Wenzl, and he is going to present on Phenomics for gene banks. Thank you, Peter.
Thank you very much. Is the mic working? Yes, I think so. So thanks a lot to the organizers for this invitation, and in particular to Philip to encourage me to talk about phenomics in gene banks. Although we hardly do any phenomics in gene banks, but I think you're right. There are entry points where phenomics can really contribute to add value to what we are doing and to improve the efficiency with which we are operating. So I'm going to talk a little bit about what we do, and then I'm focusing on where I believe phenomics or high-throughput phenotyping in general can add value and can really change the game we are playing, I believe. So we are part at CIAT of this network of CGIAR gene banks, about a dozen of gene banks, mostly in tropical regions, and each one of us conserves usually the largest collection of a particular crop or a few crops. In our case, we conserve the global collections of beans, 38,000, cassava, 6,000, and tropical forages, about 22,000. The cassava collection is conserved in vitro, and the other two collections are conserved as seeds. Over the history of the gene bank, we have distributed more than 500,000 of samples to more than 140 countries in the world. And actually, when you look at the distribution numbers of the plant treaty, it shows that the CGIAR centers are responsible for the vast majority of germplasm that is being shipped and distributed across borders. More than, or approximately 90% of all samples are coming out of CGIAR gene banks. So here's a brief overview of what I want to talk about. I'm going to first outline, I think, two areas of opportunities for phenomics in gene banks. I think one of them is really to streamline operations, like routine operations, what we have to do anyway. Perhaps we can do it more efficiently. And the second one is really enriching the collection with data, with actionable data for our users that help them to pinpoint the best bet accessions for their particular purposes. And within this value-added data domain, I think there are three different types of data. I'm going to briefly outline those, and then I'm going to enter into just talking about phenotypic data, how I view the division of labor between gene banks and gene bank users or collaborators, trade experts. I show you some examples for what we are doing in terms of characterizing the three collections we conserve, and just outline a few practical hurdles in terms of how we could really implement drones, rovers, etc., all these nice, sexy tools in our gene bank operations, and end up with one slide about a thought of where perhaps AI is going to change potentially the rules of the game, really, in terms of how phenomics could be deployed in gene banks. So in terms of streamlining gene bank operations, I think some of you know Fiona Hay. She's coordinating a community of practice involving all the CGIAR gene banks on seed quality management. And one of the topics there is seed phenotyping. And we have already seen Videometer in a different presentation. We are all the gene banks, or most of the gene banks, are now installing Videometers for seed characterization. Things like length, shape, width, pubescence. We are focusing mostly on color for beans, because this is actually a trait that's important to breeders in terms of targeting particular markets. And we have just started calibrating the machine to tell us which would be the best categories of seed color to describe the different types of beans. There are more opportunities, I think, and the biggest one for us probably is seed sorting. And we don't have an automatic seed sorter. We have about 10 people manually sorting seeds that come in from the field, a lot of manual labor that could be really automated, and we could redeploy that staff to actually characterizing the collections, adding value for users rather than just doing these routine operations. A few years back, we approached a few potential options in terms of providers of seed sorters, and nothing really worked for our purposes because mostly what's available in the market is for large volume of seeds and a few cultivars, and we have the opposite. We have usually for a bean accession, we have between 1,000 to 2,000 seeds, but we have 38,000 different types of beans, and they look very different. So you have to calibrate each accession separately, which makes it very difficult to implement any sort of automation. At least a few years back, we were told this is very difficult. Also, image analysis of seedlings to quantify germination would be a great tool to have, which we don't have. Again, we're doing everything manually. You can see on the right side all these trays with seedlings. Each of these black trays is one accession, and each of the 38,000 accessions we have to take out every 5 to 10 years to check viability. There's a lot of work that has to be done. If this could be automated, it would be fantastic, but beans are bigger than rice, so there's not so straightforward. And of course, also perhaps multispectral or hyperspectral seed analysis to predict germination, so we don't actually have to germinate. Just run seeds through a machine to predict germination would be great to have. In terms of enriching collection with evaluated data, what we are focusing mostly upon at the moment are genotypic data. Because genotypic data can not only use, we can not only generate our genotypic data for our users to sort of help them pinpoint the diversity they're looking for, but also for managing our collections to detect redundancy and so on. So that's very important for us at the moment. We're also focusing quite a lot of effort to curate the georeferencing data of collection sites so that we can then extract climate, soil, and perhaps disease pressure data from these collection sites into a database that can then be used for a variety of different purposes, for example, environmental GWAS analysis, which I'm going to show you in a moment. But of course, we don't eat genotypes or phenotypes. We don't eat genotypes or environments, we eat phenotypes. Phenotypes is really, at the end, what really matters. And that's, of course, the most complex area. And there are many possible traits. Priorities change over time. Trait expression depends on environment. And of course, complex traits such as, say, drought tolerance are really composed of a variety of different components at different levels, from the metabolome to anatomical features to root system architecture and so on. So it's really the most complex area. So diving briefly into genotypic data, that's the only slide about genotypic data I have here. We basically have 4 use cases where we use genotypic data in the gene bank to characterize the accession diversity, to understand the collection we are holding, to identify gaps, and to identify redundancy in collections. For example, we have found that about half of all cassava accessions are genetically redundant. So there's big opportunity to reduce the size of the collection and again, redeploy funds towards the characterization evaluation of the collection instead. We also use genotypic data to look at how homogeneous or heterogeneous individual accessions are. In the case of beans, most of the 38,000 accessions are land races, and land races often are populations of genetically distinct individuals. So what we're doing there is we're creating pools of DNA samples of about 30 individuals per accession, and then genotype this pooled DNA to identify accessions that are genetically heterogeneous, and at the same time estimate genetic distances among accessions. And we're just starting now to also genotype the cassava collection with a panel of markers that is being widely used by breeders, so that then our colleagues in the cassava breeding program can refer back to the gene bank collection when they are looking for new alleles at certain loci, for example, for the breeding programs. And finally, we are going to use very soon a very low-plex, cheap methodology just for ensuring that during gene bank processes, we maintain the genetic integrity of accessions. We don't mix up accessions, which is always a risk and can never really be avoided. In terms of phenotypic, in terms of environmental data, once we had curated the georeference data of the collection sites, extracted climatic values, we used this data together with genotypic data for an environmental GWAS analysis, both for the cassava and also now the bean collection, to identify environmental footprint of certain, linked to certain environmental parameters such as heat and drought. What we found were several loci where there was a clear relationship between the allelic dose of certain loci, certain genes at certain loci, changing over an environmental gradient, like here temperature and also precipitation. Based on this data, we then went back to the entire collection and pulled out those accessions that had the maximum number of favorable alleles across the loci we identified. And our colleagues in the breeding program are now using those accessions as a starting point for pre-breeding efforts. So now moving on to phenotypic data. I think there is, in general, a sort of division of labor in terms of generating phenotypic data. This is something which seems to be working fairly well in our organization. In the gene bank, we're really interested to characterize as completely as possible entire collections, ideally for inexpensive traits that are generically useful for multiple users. In terms of phenotypes, we really, like all gene banks, focus mostly on morphological and phenological descriptors for identity verification during gene bank conservation processes, not so much as these are usually not traits that breeders are interested in, except, for example, seed color in the case of beans. And if we could add some phenotypic platforms, some phenomics platforms that could collect additional data that could be useful for our typical users, mostly breeding programs, during regeneration of accessions in the field, that would be fantastic. But at the moment, we don't have those tools. And in terms of DNA sequencing, I think there's a clear path towards characterizing entire accessions, eventually at the whole genome level. And as I mentioned, the curation of the environmental data is ongoing as well, and we're starting to also acquire some near-infrared spectra as proxy for, that could hopefully be used as proxy for different traits. In contrast to us, our users are really usually taking very or relatively small subsets to evaluate those subsets for traits of their interest. So what we are usually trying to do is to collaborate with our users so that we work together, and we also eventually receive back the characterization data and can upload them to our database so that the data becomes available to future gene bank users. That doesn't always work, but that's our intention, definitely. We also are starting to try to harvest data from the internet on publications that have used accessions from our collections. So basically what we are ending up with is a sort of a collection of phenotypic data islands for different traits evaluated by different groups of users, underpinned by a sort of sea of genotypic, environmental, and perhaps NIRS data, which eventually can hopefully be used to some extent to predict some of the traits from these genomic and environmental NIRS data. That's our hope. So, moving on to show you some examples of what our gene bank users are looking for in our collections, what they are phenotyping within the organization mostly. Let's have a look here. Yep. So our colleagues in the cassava program are looking in particular, or amongst other traits, but in particular for these traits when they do their initial agronomic evaluation of any materials, early vigor, leaf retention, and early bulking of roots. And they have done a big experiment together with Michael Selvaraj's group, or the Phenomics platform at CIAT, to evaluate the core collection, about 500 different accessions in the field that's growing not very far actually from the gene bank. And of course, Michael has all these nice toys, drone-based imaging, LiDAR, and ground-penetrating radar to derive certain parameters that correlate sufficiently well with where you can construct like parameters for early vigor and leaf retention and things like that. Also, we initiated a collaboration with Mineral from Google several years back to test this robotic device, this rover they were developing. We called it Don Roberto, like for rover. And initially we thought, now let's plant gene bank accessions and throw this rover on top of them. But initially we decided to just use like a few contrasting bush bean varieties and evaluate traits that are important for disease resistance and other traits being selected for by breeders. And what we found is that, that the system is actually working quite well in terms of recognizing pods and seeds within pods. The main bottleneck really was that the whole setup had to be very standardized. So it had to be flat land, the soil couldn't be wet, and when it rains, it pours in Cali. So sometimes we couldn't drive the rover at all. And so I think it's a useful system for breeding lines, but for gene bank accessions where you have these tiny bush beans and these huge climbing beans, you can't just use one type of rover. You need something more flexible, or you need different types probably of rovers. And we were going to work on that, but then Google decided to discontinue this Mineral effort, and we were given all the images they created, and they are yet to be digested, I understand. Here's another example of what we're doing with the bean collection. We are in the process of, we have to repackage 38,000 seed pouches to essentially move our databases and move from QR codes to RFID tags. And in doing so, we use the opportunity to acquire hyperspectral reflectance spectra, in which we are not trying to calibrate those spectra with different parameters related to the nutritional traits of beans. It's starting to work to a degree, but these are just the very first examples. So I would really like to see whether we can calibrate for protein digestibility, because we know there's a huge variation in the collection between 50 to 90% of the protein being digestible or not. So on top of the protein content, if you can stack on top of that protein digestibility, you can go really very far in terms of increasing the protein content of beans. Another area we are getting involved in, I'm not sure whether you have heard about this initiative, the Periodic Table of Food Initiative, which is a big project funded by the Rockefeller Foundation, essentially a foodomics project. The idea is to go well beyond the typically up to 150 or so compounds that are usually assayed in food. I mean, proteins, carbohydrates, and fats, and within that certain types of fats and so on. But what they are doing is establishing a global network of laboratories to standardize metabolomics assays to essentially identify several thousands of compounds in each of the food samples they are processing. And initially, they're processing about 1,000 types of foods, everything from apple to Indian curry, and beans included. And in the next phase, the idea is then to focus on a few sources of food, and I hope, and I'm pretty sure that beans will be included, to look within these types of foods for the diversity, genetic diversity that could be used for genetic improvement purposes. So this is all in this area of food as a medicine domain that's happening. And finally, here's another example of what we're looking at for the forages collection. This is a new project initiated last year about the the problem that the fermentation in the rumen of ruminants of cattle produces a lot of methane, and the Bezos Earth Fund has funded this project to essentially screen the forages collections, not only of CIAT, but also ILRI and ICARDA for accessions that when digested by ruminants don't generate so much methane because of the presence of certain anti-methanogenic compounds. Tannins, saponins are known, for example, to inhibit methanogenesis in the rumen of cattle. So we are going to screen systematically about 10% of each of these gene bank collections using an in vitro assay to quantify methane production. And what we are screening for here is a low methane production, but high digestibility. So the right lower corner contains accessions of interest to us, which are then subjected to metabolomics analysis to identify eventually, hopefully, some of these anti-methanogenic compounds. Now, some of the hurdles to overcome. I already mentioned that this mineral rover worked to a degree on the main campus where everything was clean and orderly and flat, but we are working in very uneven terrain very often, and particularly for beans, we are working in screenhouses to ensure to control diseases essentially during regeneration. So what would be fantastic to have is a drone that is able to navigate within the screenhouse, knowing the spatial layout of the plots and just flying over each of these plots to identify things like diseases, because as soon as we spot a disease, we need to spray or we need to eliminate the particular accession, but also to monitor growth and to determine phenological parameters such as time to flowering and then time to harvest, pot filling, and so on. It would be fantastic to be able to have a drone that can navigate by itself rather than us manually, because that's just too complex. Now, also a very mundane problem is we're operating in very remote locations. This is one of the sites. We are growing beans in the central Andes, and any high-tech equipment that you leave there overnight is gone the next day. You can be sure. So we need actually to have enclosures to store these high-tech machinery, because it just attracts a lot of attention. And here you can see, again, referring to the rover I showed before, I mean, how do you deal with accessions like that, with bush beans like that, and wild relatives, which can grow up to 4 or 5 meters high? Very difficult. And also, I think sometimes, it's funny, but sometimes we have excessive diversity to be able, in terms of mechanizing and standardizing methodologies. Here's an example, here's a photo from one of the forage regeneration plots. And the forages collection has about 700 different species. So very different growth habits, sizes, flowering days, etc. On the other hand, the advantage of the forages collection is that we're not growing it in screenhouses. So there's room for a drone to fly and, for example, monitor biomass production, which is an important parameter, actually, of course, to our colleagues in the forages program to monitor. So if we could use the regeneration, the seed production for those accessions for the acquisition of like biomass acquisition curves over time, that would be fantastic. And in the case of the bean collection, in the case of the bean collection, I already mentioned our difficulty to find a solution for automatic seed sorting. We were told when we interacted with a group here, not here, in Germany a few years ago, who were developing such a machinery, that the 1,000 to 2,000 seeds that we produce for each of the accessions are just, it's a too small number to calibrate the equipment for that particular type of seeds. But perhaps things have changed. Perhaps there are new algorithms that can do the job. Essentially, what would be great to have is if we could run a particular accession, all the 1,000 to 2,000 seeds once through the machine establishes some criteria, cut off, etc., and then you run it through again, and you remove seeds that don't fall within the range, the acceptable range, because they are broken, they are infected, etc. So just off-type seeds. That would be really a big help. But so far we haven't found any solution. Now, finally, I think AI, and that's my hope, may change the game. And perhaps I'm dreaming, perhaps that's too far off, and it's definitely not going to happen very soon. But I'm really wondering whether eventually we can get to a point where we can use AI to screen in silico, so to speak, accessions that for a particular trait that the user is interested in, using a multiplicity of data layers, a multiplicity of omics data along the continuum from genotypes to phenotypes, like starting from genotypes to gene expression, metabolomics, perhaps certain anatomical parameters, all the way to growth habits and so on. But for that, we would essentially need for Phenomics to come to the rescue, so to speak, to help, to be able to have methods to screen large parts of the accessions for perhaps some basic sort of component traits that can be stacked on top of each other to create, to contribute to a more complex traits or agronomic performance of an accession under field conditions. I'm wondering whether this is possible. I hope it's not only a dream, but it's going to happen one day. But essentially, really, the idea would be to use these phenotypic data islands to train an algorithm to predict those accessions that cannot be screened for that particular traits from this stack of multi-omic data in the future. Fingers crossed. I don't know. Let's see. And before ending, I would like really to invite people who may be interested to collaborate with us on a range of different topics. And here's my wish list, so to speak. Really sorting out seed sorting would be fantastic. Finding better ways for viability monitoring through NIRS or image analysis to predict or to quantify germination. This could really change the game in terms of how much staff time we have to pay just to keep the wheels turning and to continue to conserve the collections, and again, rededicate that staff time to characterizing, evaluating the collections. And also, as I mentioned, like having Phenomics platforms to evaluate component traits that may contribute to heat and drought tolerance. The two big traits to screen for climate change. Everyone in our organization and the whole CGR network is targeting climate adaptation. That's the biggest topic, and I think gene banks can help if we have the proper tools to evaluate and mine the collections. Whenever we mine the collection for a trait, we find a few accessions at least that have the trait we're looking for. So if we had the proper tools available, that would be really fantastic. And again, playing around with AI, perhaps perhaps AI can help essentially to narrow down the search space. I'm sure it's not going to be perfect. Perhaps it's just 0.4 correlation, who knows? But anything that can help to narrow down the search space when you deal with a 38,000 accession large bean collection when you search for one trait. All you need is one or two or a few accessions that are really good for the trait. But to get there would be good to be able to extend the scope of what can be evaluated or predicted to a large part of the collection. That would be fantastic. And I'd like to finalize by acknowledging all my colleagues who are contributing, have contributed, and are going to contribute to what I showed. I mentioned already Fiona, who is managing the seed quality management network. Luis Guillermo, Monica, and Juan Jose are part of the gene bank team, working on bean conservation, forages conservation, and the molecular characterization. Tatiana is working with Darren Drury from Ohio State University on the field spec, the hyperspectral seed reflectance project that I showed you a few graphs of. Jacobo, Alejandra, and Andres are working on the project to identify low methane forages funded by the Bezos Earth Fund. And that's it. Thank you very much for your attention. I'm happy to take any questions.
Peter.
Thank you very much, Peter. That was a very enlightening talk to give a little bit of insights into the application of plant phenotyping for gene banks, besides the traditional applications in conventional plant breeding and in, of course, research. I think that opens our minds to all the further applications that there are and the possibilities that plant phenotyping can open up and enrich. We can have one or two small questions, and the rest then...
The big ones for later?
The big ones for later, exactly. We'll have more time. So are there any questions right now? Immediate questions?
Yeah, thanks. And sure, I'll have a longer question.
Okay. Well, perhaps just one long one instead of two short ones.
All right.
But can you, instead of characterizing the seeds, can you characterize the users of the gene bank?
Can you characterize?
That's not a bit of a joke, but the users of your gene bank.
Mm-hmm.
Can you describe who are the main users and?
Yeah, the main users are national research organizations in the global south, but we have a lot of users also in the global north, mostly at universities, and also universities in the global south. So these are the external users. So when we distribute outside of CIAT, and internally we distribute a lot to the breeding program. So there's a bean breeding program, a cassava breeding program, and a tropical forages program. And so there's a lot of internal germplasm flow, but the other ones are mostly universities, national research organizations like USDA in the US, for example, as well, and universities.
Yep.
Okay, thank you very much. One more applause for our keynote speaker, Peter Wenzl from CIAT.
Thank you.
And as the first speaker of the session, I would like to call to the front Dr. Koji Noshita from Kyushu University. Welcome. Dr. Noshita has been working on morphometrics and computational biology or mathematical biology. His talk is called Network Feature-Based Phenotyping of Leaf Venation Robustly Reconstructs the Latent Space with Safe Seeds.
Thank you. Thank you for introduction. I'm happy to be here. So I'm really interested in the morphological aspect of the organisms. So then I developed the theories and the techniques to quantify the morphological property of them. So today I'd like to talk about the morphological diversity and the several constraints on the leaf venation patterns. So I expect all of you guys have seen the leaf venation, leaf vein structure like that. So then the leaf venation of the angiosperm, especially the dicot, exhibit the hierarchical, the reticulate, the structures. So, then the first to third relatively thick veins called the major vein, and the fourth to the higher order relatively thin veins called the minor veins. So, and these leaf venations are also considered as the very important characteristics for the taxonomic classification. And some craze showing some specific venation architecture. So, and the leaf venation it is the transportation network of the water, the nutrient, and the chemical signals, right? So then the people believe that the leaf venation structures contribute to the various type of the physiological and the ecological functions. So, for example, as I say, the leaf venation is the transportation network. So then, the hydraulic conductivity through the leaf it strongly correlate to the leaf venation structure. So, for example, the vein length per unit area (VLA), as known as the leaf vein density, is known as the correlate with the leaf hydraulic conductance. So in addition to that, under the natural condition, the leaves are often damaged by the physical perturbations and herbivores. So then, some researchers reported that this venation pattern contribute to the tolerance of the damage. Then it means that if they have some passes, alternative passes behind the damaged region, it's beneficial to the survivorship of them. So in other words, how can I say, if the venation pattern have the detour, it's good. Oh, sorry. So then I believe that this venation pattern should be quantified as a network. So however, most studies evaluated the leaf venation patterns based on the relatively simple measurements, like the length or diameter or branching angle of the main major veins. And as I say, the VLA or vein density. And sometimes it's also used the areole, which is the smallest area of the enclosed by the veins. It is used for the index of the degree of the loopiness of the venation patterns. So is it enough? Are they enough for characterizing the whole venation patterns? So I believe that we need another aspect of that. So then, recently the topological approaches have been proposed. So I think that this is the promising direction because the vein structure is intrinsically represented as a network. So then in this study, we firstly developed a simple high-throughput phenotyping framework for quantifying the vein network based on the specimens. And then by using this method, we investigated the morphological diversity of several constraints of the leaf venation network. And finally, we identified the latent space of the leaf venation pattern. So firstly, I'd like to explain the framework of the purpose in this study. So in this framework, we assume the two types of the data as an input. So one, the untreated image data, leaf image data, is just digital data, digital images of the leaf data. And the other one is the cleared leaf data. So traditionally, the cleared leaf data are used for the observation of the anatomical detail, anatomical structure of the leaves, including the venation and the vein structure. So based on from these image data, we firstly segment the leaf venation area by using a segmentation model, then extract the graph representation of the venation pattern. Then finally, the network features are calculated from this undirected graph. So then it means that we can represent the one venation pattern on the leaf can be represented as one vector, right? Then, we can enjoy on the vector data, any types of the statistical method. So let me explain the more detail of my data. So first one is the untreated leaf data. In this study, we collect about 500 leaves of the five species in our campus. Then I just put the leaf material on this light table, then capturing the image data by digital camera with the specific frame, which have some markers. We can collect the distortion based on these marker information. And if you need, we can introduce some size information by using these kind of the markers. And another one is the cleared leaf data, cleared leaf image data. So we use this open public data set, and we make several types of the subset of this cleared leaf image data. One, the full quality image data set. It is used for the training the leaf image quality recognition model. So then, and we also make a high quality data set. It's used for the training the segmentation model. So, and finally, we also make a cleared leaf data, which is consistent of about 300 good quality leaf image data of the 5 genus correspond to these 5 species. So by using this image data, firstly, we make a segmentation model using a unit. So the point is that we use the high quality data for training the unit model for avoiding the annotation cost. So yeah, could you imagine the effort on the manual annotation of the leaf venation pattern? It's one of the nightmares for me. So then we firstly select the good quality leaf venation, cleared leaf image data, then just applying the conventional image processing to make a mask correspond to the original images. Then, based on this training data, we train the unit model. Then the trained unit model can estimate the segmented leaf vein images from both the untreated and the cleared leaf data. So from the segmented images, we extract the undirected graph representation. So I skipped the detail, but I briefly explain the process. Then firstly, we extract the topological skeleton based on the segmented images, then extract the endpoint and the branching point as a node of this graph. So finally, we connect these nodes with the edges. So we make this undirected graph representation based on the sample data. So finally, we calculate the network-wide scale invariant features called a net CIMMYT. So in net CIMMYT, the 7 types of the node features are calculated on all of the nodes, right? Then also the summary statistics of these 7 features are calculated. Then finally, we got the 28-dimension vectors as a representation of this kind of the venation pattern. Okay. So that's our proposed method. So let me show you several examples of each process. So firstly, these are the examples of the segmentation and the undirected graphic representation. So as you see, I'm not sure, resolution is limited, so then, but we successfully extract the leaf vein lesion from the original images, like on both of the untreated and the cleared leaf image data. And based on these segmented images, we also extract the undirected graph like that. If you want to see the details there, please come to me after the presentation. So then, I will show the detailed structure of that. So, and finally, now we can calculate the network features based on the undirected graphs. So I'd like to demonstrate that these network features have enough information to distinguish these species based on the only network features. We don't use any kind of the information about the texture or color or something like that. So here we just use the random forest model to recognize to classify these 5 species from the network features. Then in this analysis, the random forest model can classify the 5 species with about a 90% accuracy. So it means the network features have enough information for distinguish these 5 species. It's just a demonstration. So then, let's move on to our main result. So that one is the summary of the network features calculating based on the untreated leaf images and the cleared leaf images. So then we just apply the PCA on the network features of the untreated and the treated cleared leaf images separately, right? But interestingly, we can observe some characteristic distribution, one-dimensional distribution pattern in both of them. Yeah, these, how can I say, the PC space are, reconstruct separately, but we can find basically the same pattern. What is that? So to understand this distribution pattern, we put crop the segmented images instead of these markers, like that. So could you see this region? So then we can find a very interesting pattern. Here, around here, these higher order veins showing how can I say, the open tree-like structure, like that. And in this region, we can also observe the closed loopy structure, like that. So, and the higher order venation pattern are gradually changing along this line from the tree-like structure to the closed loopy structure. But why? So we found a very good previous study. In this study, the authors explore the optimal network structures based under the 3 functional demands. One is the transportation efficiency, and second one is the construction cost of the network, and the third one is the robustness to damage. They try to find the optimal network in these 3 functional space. Then they finally found this red region as a palette front of against these 3 functional demands. So palette front means that a subset of this functional space which cannot improve any functionalities without degrading others, right? It's one of the, auxiliary. These are the palette front. And along this palette front, they also found that these network patterns are changing from the closed loopy structure to the open tree-like structure. Maybe we found basically the same latent space on venation patterns on the leaves. So these 3 functional demands are basically consistent to the functional demand of the ecological or physiological functional demand of the leaves. So then, we can find basically the same pattern, like one-dimensional U-shape distribution pattern, and in this one-dimensional pattern, the veins, higher order veins showing the open and tree-like structure to the closed loopy structure. So maybe the plants or venation patterns occupy this specific subspace of the phenotypic space through as a consequence of the evolutionary process under the several functional demands. Okay, so in this study, we developed a simple and a high throughput phenotyping workflow for quantifying the vein network and identify the leaf venation specific morphospace. And these patterns maybe correspond to the palette front of the optimize the transport efficiency, the construction cost, and the robustness to damage. So I would emphasize that these latent space, we can identify that these latent space because we quantify the venation pattern as a network, not simple, just a simple measurement. So I would like to say that, yeah, we need to use appropriate model or representation of the target for understanding the phenomena or some structure you're interested in. So thank you. That's all.
Thank you very much, Koji, and I think we have enough time for some questions.
Hi, it was a really interesting work.
Thank you.
So I have a question. Because you are using the graph topological structures to do this latent space, but are you using any features? For example, in each node of the graph, you might have an X and Y position that will tell how are these nodes positioned inside the leaf, or are you using only the topological general information?
Yeah, I think we can use such a kind of information in addition to this purely just only on connection information. But in this study, we just use this network, undirected graphs representation, excluding the other types of geometric features like length, width, or angles, or something like that. But yeah, I think we can add such a kind of information into our analysis.
Thank you.
Further questions? If not, then I have a question.
Sorry.
Please.
Oh, yeah, sure.
Okay.
Yeah.
In the graph that you showed for the PCA, first two axes, there's in the central lower graph, there's a strong horseshoe kind of pattern that you can see. Have you checked maybe using a detrended principal component analysis if this is an artifact, or is that really derived from the data?
So you mean what kind of the artifact?
Yeah, typically when you see in PCAs these kinds of horseshoe bow effect, that could hint at the data maybe not being optimally suited for PCA.
Thank you. It's a very interesting point. And we check, not check the artifact or not, but we also try to find out which network features correspond to the PC1 or 2 or 3. So then we found that the PC1 and the 3 space correspond to the kind of how can I say, the number of the neighbors of the EGONet. It is one of the one-hop subgraph. So then it means that it can capture such a kind of the closed loop or open structure by this PC space. So and we also found that PC2 is a kind of a noise of the images. So then maybe we can understand and interpret each PC space, PC axis related to what kind of network structures. So then I believe that this structure is not artwork on the Thank you.
Thank you.
And if I had time, so just I'd like to advertise our other work. So we developed several types of the method to quantify the very diverse morphological properties. So today I talked about leaf venation pattern, but we also try to developed a method to reconstruct the 3D leaf edges and the 3D surface structure of the potato. But maybe this approach can be applied to the any kind of the 3D surface structure, like a fruit shape or something like that. So, and we also try to optimize the process for getting the 3D information. So then, some members come here and provide a poster, so that please discuss with them. So thank you.
One more round of applause for Dr. Koji Noshita. Thank you very much, Koji.
Thank you.
It's been a pleasure to have you. And as our next speaker, I'd like to call to the front Dr. Harkamal Valia from the University Nebraska-Lincoln's Department of Horticulture and Agronomy. His talk will be on spatial temporal dynamics of heat stress response in rice using 3D reconstruction.
Thank you, Philip. Thank you for the invitation. All right, good afternoon, everyone, and welcome to Lincoln. I'm a local person on the committee. So I'm a plant physiologist, and my interest is in understanding how crops, especially cereals, respond to abiotic factors. The two primary factors that I work on are, while evading a fly, the heat stress as well as drought stress. So today I'm going to talk about some of the work we have done in terms of looking at the dynamics of how crops or a plant responds to a particular stress. So the dynamics, I mean both the temporal as well as the spatial dynamics, with the idea being that the plant typically for most stresses would not respond uniformly and would not respond in the same way consistently. It would evolve with the stress and the time. So the work would be on rice, and the idea that I want to pursue is higher temperature. As you can see, this is the historic data for how the temperature anomalies have played out at the daytime highs as well as the nighttime highs for average. So in red is what you would call is like higher than the average or temperature, so anomalous temperature, and the areas that are blue are the ones that typically have kind of been colder than what would be like average over the almost 100 years. The two key things I want to point out here is that there's a lot more nighttime increase in temperature as opposed to daytime. So because there's a lot more red that you see on your right as opposed to daytime. The second thing that I want to point out is the daytime highs seem to be very speckled or sporadic in terms of spatially, whereas the nighttime increases seem to be more widespread and more continuous. In the US itself, in the last 100 years or so, the nighttime temperatures have increased at twice the rate of daytime temperature. So understanding how plants are specifically responding to nighttime temperature is quite important. The crop that I'm going to talk about is rice. These are a little bit dated, but they show what in terms of kilocalories per capita is consumed globally. So if you combine rice and wheat, it collectively feeds more, provides more than 50% of the total calories that we consume as a species. So this was data from 2004 from field experiment led by the International Rice Research Institute, which showed that for about 1 degree increase in the nighttime temperature or the average low temperature during the growing season, you could get up to about 10% decline in the yield. So the matter is not only about the yield, which certainly is very, very important, but it's also about the milling quality. So rice needs to be polished, and if you have milling issues or quality issues, then the quality, in terms of breakage and usage, you have poor rice, and a lot of it can get wasted. and not consumed by humans. Similarly, hybrid rice, there's some evidence that suggests that hybrid rice, which is other type of rice that grows a lot in the US and in many other parts of the world, is also more susceptible in terms of quality and yield to higher nighttime temperatures. For my study, I'm going to call high night temperature, since it's a long word, HNT. So from a physiological perspective, we decided to focus on the reproductive stage. As the temperature increases at nighttime, the rate of respiration increases. So plants start to lose more of the carbon. During vegetative stage, there's ideas and some evidence that plants are able to compensate because they're actively photosynthesizing, and they've got more carbon than normally they would have during reproductive stage, where they're trying to actively store. So it also not only you lose carbon, but the carbon that is packed, which is starch, in primary form of starch in the rice endosperm, its rate of accumulation and how it is packaged is disturbed, which means that the seed actually becomes a little bit weak. So during milling, it could break more easily. So when we study rice grain quality or chalkiness, we talk in terms of the rice panicle architecture. So there are grains that are pretty nice and plump, and that are called superior spikelets. So these are all branches, and these yellow blobs are grains or spikelets. And then there are grains that look a little bit half-filled, not so small, and like a little bit off types. So these are called inferior spikelets. So that creates a pretty interesting challenge. You want a lot more superior spikelets, but higher nighttime actually gives you a fairly decent, significantly more inferior spikelets. Given that, what we wanted to explore was to characterize this response, but then do it for a large number of rice varieties from a diverse germplasm that represents a diverse germplasm. And the reason for that was that we wanted to understand if there is, what's the genetic basis of this response under high night temperature when the grain is developing in those conditions, so that we can come up with genes and markers that could help improve that trait. So to do this, we set up an experiment, to do this, we set up an experiment where we took about 300 rice accessions from one of the several diversity panels that people use in our community called the Rice Diversity Panel 1. It represents about rice from all major subpopulations as well as from 80 countries. So we used this and we grew the plants under controlled condition in a greenhouse, and then depending on when half of the primary panicle, which is the first panicle to emerge, is mostly flowering, we mark those panicles. We wait for a day, because that's the day when we mark it. When it looks like this is the day when it's fertilized, we let it go for one day, and then we take a subset of our plants from that particular accession, and we continue to grow them in controlled conditions. And we also take the other part into a high night temperature greenhouse where they mature under that condition. So essentially, it's 30-degree daytime, 23 for night, and it's 30-degree daytime, but the night temperatures are increased by 5 degrees. So between 4 and 5 degrees, given the greenhouse's control. And then what we do is we wanted to capture the dynamics of the panicle development. So we imaged the panicles 3 times. So week 1, week 2, week 3, depending on when they fertilize. And it's a diversity panel, so it goes anywhere from 2 months to 4.5, 6 months, but we capture the reproductive stage. This is what the animation looks like. Essentially, we built a box from the local hardware store, have cameras, and a desk height adjusting mechanism from Amazon. So you could take the plant depending on its height and image this panicle. And the idea is that we should be able to capture the phenotypic variation to identify eventually superior spikelets and inferior spikelets, which obviously are not in a distributed. So there's a lot of heterogeneity from that perspective. So this is what the real thing looks like. The rest of the plant is underneath. So we just have the panicle, and there's cameras that go around at different angles. And we use these images then to make a 3D reconstruction. So essentially, if you want to look at this, we have week 1, and we have week 2 and week 3. So as it progresses, the volume changes, the surface area, and the color, all of those things change. So we used, let me see if the animation would work. So we used the NERF software to make 3D reconstruction. So this is not an image, it's a 3D reconstruction, and it comes out very clean. So we can use this to capture a lot of phenotypes, like related to color and volume and morphology. One instance to show for a handful of genotypes, this is just the ratio of green pixel to red pixel, which would kind of be indicative of maturation. You could see that they're week 1, they're kind of similar. Maybe the HNT, which is the light gray box, is kind of exceeding, but not significantly. But then you see that progression, so it advances much faster and then looks different at the end of week 3 at the whole panicle level. So we next asked if, since we have this panel and it's heavily genotyped, so we have about 1.3 million SNPs for this, could we then do GWAS and see what do we find? So we used, we used the week 1 data for just the green pixels. And one of the major peaks, these are just 12 chromosome of rice, and that kind of caught our attention was a SNP that lies in the in a gene called SPED1. Several years ago, a group, Jiang et al. Had shown that if you mutate SPED1, you get these very small branchings or rachis, so that creates a very compact panicle as opposed to a wild-type panicle like this. So this kind of showed us that what we were observing is actually, capturing the genetic variation, and there's like a, validation for what we are doing. So the next thing we wanted to ask was, we were interested in this superior and inferior, and we know that they're not uniformly distributed. So could we actually segment? So if we have a 3D volume, like a 3D point cloud structure, could we actually segment or slice it into 10 geometric segments and then treat each of those segments as a independent or as a trait? And could we then analyze that to see if we could find the basis for this, differences that may be spatially, correlating? So to do that, so just to give you an example, this is what it looks like in week 1. So it's very compact, not filled. As the grains get heavier, they get more plump. They start to spread out a little bit more, and then eventually, as they mature by week 3, they are like changing color. So this would represent the red to green ratio that I was talking about. So we did that for the phenotypic data, and we also ran a genetic correlation analysis for these using the red pixel count for control and HNT stress. So this is a heat map which shows that what's the correlation between each of those traits. So if each of those slices next to its next slice and every other slice of that panicle. So what we find is that there's definitely some strong correlations. If you have a positive correlation spatially, you would see this in red, and if it's a negative strong correlation, you would see this in blue. And what we find is that the traits or the segments that are next to each other are genetically correlated. What that means is that there are genes that are commonly shared in regulating that particular trait. In this case, that trait being a digital trait, basically a red pixel count for that segment and the next one. We also found that the controls have a lot more well-partitioned. So there's a clear middle and there's a clear bottom and a top part, because these are kind of correlated, these are negatively, and then these are again positively correlated. So that seems to erode under HNT stress, which is to be expected because I think as the stress continues, the HNT accelerates the maturity of the panicle, so you would expect something like this. So this analysis was done by my collaborator at Virginia Tech, Gota Marota, and his lab group. So with this idea, we actually did GWAS for all the normally distributed characteristic that we could find for different pixels and ratios and volumes. And so there's an instance of what one of the results looks like. So you could have, in this case, you could have a strong peak at in segment 4 and segment 5 and 6, but not in 7. So that shows that there's a genetic basis for how the panicle is developing in the different temperatures that has a spatial basis to it. So you may not see it if you were to just image the top of the panicle or the bottom. We ran this analysis, so I want to give you, and we found several very significant SNPs. So I'm going to give you a snapshot of what we did with one of the SNPs. So this is what we call HNT 5.1. So this is the hist, the third segment, and our red pixel under control. And this is the third segment from the bottom, and the red pixel under HNT. As you can see, one of the major peaks here, which we call HNT 5.1 on chromosome 5, has a strong signal here, but it's missing completely from control. So we investigated the gene, which is in proximity of this particular SNP, and we find that this gene is not characterized in rice, but it has a very strong diurnal regulation. So I want to pause and kind of mention that plants perceive day and night through light and temperature. So if you make the nights warmer, you would think that it would perturb some of how they perceive the beginning of the day and night. So so this gene actually peaks quite strongly around 4 a.m. and 6 a.m. and then it kind of creeps down, and then it starts to go back up and peaks again. So this is from a public data set collected in the field over a development window. So we selected one of this gene, and we made gene edits. It's a FBOX gene, which is like there's several hundred members of this gene family in rice and in other plants. And we called it FBOX 5, lack of any creativity there. So what we did was we made several edits, and I'm showing you data for 3 of them. When we grew them, after a couple of generations, we find that the panicles were shorter compared to the wild type, which is shown here as KIT, and the mutants, the edits are shown here as PY and different. So there are different types of mutations to make these 3 mutants. We then imaged them, these panicles, under control condition and under HNT condition in week 1, week 2, and week 3. We detected this SNP on week 3 panicles, and what we find is that the only place that we see a difference in the R to G ratio is in week 3 and under HNT. So if you look at just the controls at week 3, they're not at all different, at least not very significantly. But if you look at an HNT, you have a wild type, and then you have these mutants. So they're kind of diverging, and the divergence is kind of starting around that segment 3-4 area. So that kind of is one example, and we have one more of that. But we wanted to explore this a little bit further in terms of, we don't know anything about this gene, and we think it's kind of important. So we looked at the expression of the mutants, 2 of the mutants, and the wild type at 4 a.m. and 6 a.m., red being the HNT and blue the control. We see that this gene is suppressed in the mutants as well as the wild type by HNT conditions. When it's supposed to peak, it doesn't peak as much under high night temperature conditions. So when we looked at the other genes in the genome through RNA sequencing, we found that some of the major clock regulating genes like TOC1 and PRR37 at Jagentia and another clock gene, CCA1, they are strongly disturbed by this mutation. So the first half is basically the wild type, and then you have the mutants at 4 a.m., and then you have the wild type again at 6 a.m. and the mutants. But at 4 a.m. especially, these two genes that are sort of central for how the plants know that it's daytime versus nighttime. So this gene seems to be upstream of the clock, of these clock genes, and obviously their regulation is disturbed when you look at the second half of this 4 a.m., 6 a.m. under HNT conditions. So, that kind of gave us some new clues to how the warmer nights may be regulating the high night temperature response. Just kind of before I conclude it, I know it's a gene bank talk. So the different allelic forms, which is like the major and the minor. If you look at how the 300 lines are distributed, or the ones that have one form of the allele or the gene globally, this is what it looks like. It goes all over pretty much. But if you look at the minor alleles, their distribution is mostly at high latitude. There's hardly anything in the major, some of the major rice growing regions. So that sort of suggests to us that this gene is actually important for potentially like latitudinal regulation, and that higher nighttime temperatures. When we think of higher latitudes and lower latitudes, we think mostly in terms of light and photoperiod, but perhaps the nighttime temperature is also a factor in there. I know my time's kind of running, so I just want to summarize a few things. One, that you have conditions, we can screen for stress tolerance, but there are loci that are very unique, as I've shown, to the stress conditions. And if you're screening germplasm and material under optimal conditions, you may not detect them. So this would be like one or at least a third instance that my lab's kind of shown that in case of temperature. I also want to point out this idea of 3D reconstruction. I think not that I'm pointing out, I'm reinforcing the idea from the talk, the talk before mine, and several other talks and posters, that, this 3D is not just like a fancy, oh wow, look how nice the pictures look. There's actually real potential to understand the dynamics and variation. And so, there's a lot of promise here. And finally, I think we've looked at this gene in corn and sorghum as well. It's not characterized there. It also has a very strong circadian regulation. So we're starting to study that to see, if we could find more evidence for its adaptation in terms of wider germplasm. And with that, the most important slide is, if it would advance, I want to thank my current and past lab members, especially Anil Chandran. This won't be possible without my computer science collaborator at UNL, Hong Feng, and a lot of work was done on the genetics side by Gota Maroda at Virginia Tech and his lab. And with that, thank you. And if you're interested, we're looking for a postdoc who could work in this area. So please feel free to reach me. Thanks.
Thank you very much, Kamal. I want to open the discussion now. Any questions? Everybody's looking forward to the coffee break. No, we have a question here.
Great. Thank you for your great talk. I didn't know even that such small variations in night temperature could have such an impact on the physiology. So I was thinking if did you do any typical physiological measurements at the beginning of the day, like stomatal conductance, photosynthetic efficiency, to see how those plants get up?
Yes. So in terms of the conventional phenotyping, we did measure the grain parameters, so grain length, grain width, grain weight, and we found and validated the genes for those. none of those hits match with what we are seeing with this dynamics. In terms of the physiological measurements, it would be very difficult to do this for several, I mean, we're talking 1,800 plants. So it would be very difficult to kind of have a physiological traction for doing this on a dynamic basis. But what we did do is like we took some of the mutants and wild type and some of the diverse lines, and we are looking at the, or we're doing experiments where we can look at the panicle level gas exchange under different temperatures. So we have a customized cylinder that we built with Leica, and we combine that with the imaging data so we could get the surface area to normalize, because every panicle is going to be different. So we can attach a 6800 to that cylinder of living plants and have different temperatures. So we are doing some of it, but that's just completely intractable as far as I can envision with like a genetic diversity site. But yeah, but I'd be happy to show you what we have so far. That's a nice question.
Thank you.
Thank you so much for the talk. It was quite interesting.
Thanks.
I was curious about the 3D reconstruction. You said that's a very important new tool. And what I wanted to ask was, and you used it in a way I hadn't seen done before. I was curious what you saw as being like the future of 3D modeling and what are some things that you would like to do in the future with it?
Okay. I was wanting to show that in this presentation, but somehow it did not update on my, so we built the system. Essentially, the whole system with cameras is probably going to be cheaper than the 4-night stay in this hotel. The new NVIDIA software called NERF, if you look it up, would not require the 120 images we use to analyze this. So that software can do a much faster job on a less computing heavy setup with a simpler camera and maybe 3 images rather than this whole, 1 a second from 2 cameras. The second thing I wanted to highlight, and I could show that, sorry about this update, but my colleague Hongfeng Yu developed a graphical interface for this. So that interface, he's going to basically he's fine-tuning it, but essentially you can upload these images and it can analyze, and then you can look at different segmentations. You can track different branches over time if you have the images, and branch length and seed number, and individual seeds can be tracked for basically a month. And in terms of the gene bank enhancement, that's like 500-600, and an open publicly open source software that we are developing and want to upload fairly soon once they publish it in a conference. Computer science people do conference publications. So once that's done, I mean, it's a powerful tool and very cheap, as long as you have hands to work through to kind of be gentle with those plants as you grow them, whether it's under control conditions or not. So yeah, so that's definitely maybe I rambled off your question a little bit, but I wanted to say that because I had it, but it didn't update. But yes, I can show you if anybody else is interested. I have an updated copy of my presentation on my computer. So
I want to close discussion here now because we are running a little bit behind schedule. So in order to allow a little bit of a coffee break, I see you back here in about half an hour. One more applause for Harkamal.